So people tell me if any questions from the previous sessions. Any type of question, any doubts, anything that you want to clarify from the last session, or maybe prior to that also. No questions? No questions, then we can proceed with our session, right? Yes or no? Shall we proceed?
Yes.
Okay. Chalo then. So till now we have covered in section 16, we have covered 16.1, 0.2, 0.3, everything, right? So now we will start with section 16.4, correct? So what is section 16.4? Anybody?
Characterization of risk.
Yes, characterization of risk. So what do you people understand by this characterization of risk? Let me share my screen, then just let me know once you people are able to see my screen. It is visible, right?
Yes.
Okay.
Visible.
Chalo, today we will discuss about characterization of risk. So what do you people understand by characterization of risk?
To
Anything?
confirm whether it is important or not.
Okay. What else?
The trends of the risk, like the population getting affected, then the public health impact,
Mhm.
uh,
Oh,
reversibility.
right. Okay, great. So see, something will happen like suppose if we are describing something about a person, like let's take an example of you guys only. If I want to say that, suppose like Shahrukh has mentioned, so who is Shahrukh? So there would be certain characteristic related to Shahrukh, who is Tanmay, who is Amrita. So there would be certain characteristic to you people, okay? In the same manner, the risk has to be categorized based on their characterization. Means we characterize the risk to understand as to what the nature of that risk is. Means like we studied in earlier sessions that we categorize it into important identified, important potential. We keep it as missing information also. So in characterization of risk, we will try to identify those parameters based on the information from those parameters. We will categorize the risk as whether it is important identified or important potential. So these are the parameters which are usually used to categorize them. And when the risk is identified, like suppose, don't think it from the generic perspective. Think it like the molecule is just approved for marketing. It is just a new market. It is just come out of clinical trial and the NDA approval is done. And now the molecule is into the market for post marketing. So at that time, you won't have much information about the molecule or the risk also. Like first RMP, when you are preparing at the time of approval, you guys must be knowing, or if you are not knowing, then I'll let you know that when we complete the clinical trials and approval, we file the approval, NDA approval. At that time, there are many documents that are submitted to get like the clinical study reports are there. There are the CTD molecules that are submitted. CTD module, CTD is submitted. So with all these, we also submit our RMP. That is a mandatory requirement. So in the RMP at that time, if we have identified some risk is there, so we will present the information about that risk into the RMP. So just a minute, allow me 1 minute. So at the time when we are making our RMP for the first time, and there are certain risks. So we might not be aware of all the characteristics of that risk. So in that case, we may go for past studies to characterize it, as we learned earlier that additional PV activities are mostly they are conducted to further characterize the risk. So maybe we can go for past studies. We can go for observational studies like pregnancy registry, some serious diseases registry, and all those things to get the information on the risk, emerging risk, so that we can characterize it further. So I'm talking about like the first time when we are doing it. So we would have little information about that risk, means its characterization parameters. And eventually, as the molecule grows into the market, gets older into the market, we get more information, and the same information we keep on adding into the PBER or the RMP, so that we will reach a time when we will say that we have fully characterized the risk. So till now, were you able to get the basic concept? What is the characterization of risk? Why do we do it? Right? Any questions till now? Till here, don't ask about the parameters. Parameters we will discuss slowly. So till now means what I wanted to convey, like what should you understand by characterization of risk? Nobody asks you that what is the characterization of risk. When someone asks you characterization of risk, you have to explain those parameters only. But I told you to understand have the basic understanding of the characterization. Why do we do this characterization for any risk? Any question till here?
I have one question here. In RMP, we are getting the information regarding the characterization of risk. Means we are submitting that there is a potential risk, identifiable risk, and missing information. Okay, so there is the source document for that is the clinical technical document CTD is there. But I have a means I have a question that in the same information, if we are submitting the first time PSU February, then the same information we have to copy from the RMP, or we have to means create new one here.
See, what happens if your RMP is there for the molecule? Okay, you are writing the first fiber. You have the RMP in place. In RMP, whatever risks are there, so you have decided with your physician that the same risk will go into the PBER. Okay, then whatever characterization is provided in the RMP, same you will copy paste in the PBER, which you are presenting for the first time. The process is follow throughout. But what happens when you are going for certain interviews or something, they will ask you, like they will put you in place that suppose you are writing the first RMP only, or suppose you are writing a PBER in which there is no RMP. How will you characterize that risk? There is no source to copy paste, right? There is no RMP. So how will you characterize that risk? So we will learn about those things in today's class. You are correct that if there is an RMP and the same risks are going into the PBER, the characterization will also be copy pasted from the RMP. Okay?
Yes.
Did you get your answer?
Yes.
Okay.
So mostly it is for new molecules, right?
No, it is not for the new molecules. Every PBER you will have to write the characterization of the risk. I just gave an example of new molecule to make you understand as to what characterization is. Characterization of risk is done for every molecule, whether it is 15 years old into market, 1 year old into the market, or 30 years old into the market. There might be one scenario that suppose a molecule is well established, it is fully characterized and everything. So some clients prefer to say that there is no change in the characterization of risk from the previous one, from the whatever is known till date, and they write that sentence and close it. But ideally, characterization of risk should be provided. The table should be provided for each molecule. So you have to remember that it is provided for each molecule. You don't have to go by that. If you work for some client and they are doing like that, then you have to follow that. But ideally, you should remember that you have to characterize the risk in 16.4.
Yeah, but for established molecule, the previous PBER or RMP is there, right? So we can copy paste from there.
You can copy paste from there. That is your part of working. You will copy paste, no problem. But if it is not there, in this class, we will learn as to how we are writing the characterization for the first time.
Okay.
Any other question?
So apart from past study, any other pharmacological activity we do? Observation study, disease registry, and other than that?
For specifically characterization, we do these studies only. Apart from this, so I'm not aware. I'm also not aware that we are doing anything. Normally, whatever cases we get in ICCR or whatever new clinical studies are being completed for our molecule, those are the sources apart from this past studies, observational studies, and all. I'll discuss about the sources because sources are more important in this section. People ask about what is the source from where you are taking the information, how you take the information, and all this.
So it is for the risks for which the characterization has not been done yet, right?
No, 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 no. Don't confuse with that. Characterization has to be provided for each risk which you have listed in section 16.1. Section 16.1, you remember summary of safety concerns. The same you have
Right.
presented in section 16.3. So whatever risk you have listed in section 16.1, you have to characterize all of them. Characterization mainly is done for important means identified and potential risk. Missing information, there is no such parameters of all the parameters which we will discuss. Those are not followed for missing information. Missing information, I'll let you know how we present. But characterization is for each risk that you have categorized as identified and potential, important identified and important potential, that has to have a characterization table for them in section 16.4.
Sir, what is past study?
Post-authorization safety studies, we discussed like there are additional PV activities that are ongoing. Okay. So after post-authorization, what does the mean? That your marketing approval is done, your product is into the market. After that, whatever safety studies you are doing, those are called as post-authorization safety studies. It might be observational studies also, which are non-interventional. There might be certain interventional studies also, which are initiated after the marketing to study the safety analysis. Because what happens sometimes when we conduct the clinical trials and complete it, we don't have much information about the safety, but there are certain risks which are anticipated. So to characterize those risks, because we don't have much we didn't got any cases in during that, but we know that those risks are associated or maybe are important identified with that. So in further steps to characterize those risks, the MAH itself plans, or it might happen that certain times when giving the approval, regulatory authority may ask you to conduct the pass study for this respective pass study. Like they could say that you should monitor the patients who are uh, who are having a history of cardiovascular disease and what is the safety profile emerging in them. So in that case, you will plan a pass study in which you will enroll those, means you will monitor the uh, drug prescription and the drug being taken by the patients who are cardiovascular uh, disease patients or having a history of cardiovascular. So this pass study will be conducted by you, and whatever information, safety related information or whatever important information related to characterization is emerging from that, you will document into your further PBIRDs or RMPs as it is evolving. Okay.
Sure.
Anything else? Anything else? People? If not, then we will move with next part. No questions, right? People, no questions. I'll move forward, right? Chalo. First, I'll list the parameters that we. So, and then we will go for each parameter individually. Potential mechanism and the frequency, seriousness. Seriousness, you can keep with seriousness, you can keep outcome also. Then in some we have severity also, but leave it. No problem. Then risk groups. Risk groups slash. I chose to write in front of you so that you people can remember. Risk factors, then preventability, then preventability goes with reversibility. You can reversibility, then impact on patient and public, we can say both together. Individual. Then we write evidence source, strength. So see, I have mentioned here 7 parameters basically: potential mechanism, frequency, seriousness, outcome, RS group, risk factor, preventability, reversibility, impact on individual and public health, and evidence source of strength. So what happens in certain companies, you will get seriousness, outcome together, and there will be one or more factor, that severity of the risk. But since if it is no clinical trial, then you won't be able to find severity of risk, so you can omit it, no problem with that. Then some companies bifurcate this in preventability and reversibility in 2 parts. Some bifurcate this impact on individual patient, impact on public health as 2 parts, but no need to do that because both all are same. So now ideally you have to remember all this. Okay, are you getting it? What are the parameters on which we characterize it? Potential mechanism, frequency, seriousness of the risk, risk groups and risk factors which are more prone to have those risks, preventability of reversibility. Preventability or reversibility means how we can prevent, or if it happens then how we can reverse, impact on individual patient and impact on public health, and then evidence source and strength. Evidence source means here it means that how we got this information. So we will discuss each and every section step by step. Chalo. So first we will discuss as to what is potential mechanism. So like suppose if I take an example of anemia, our risk will be anemia. Okay, it is an important identified risk or important potential risk, whatever you take, there is no differentiation. We have to characterize them in the same manner. Chalo, we will take anemia as an example as a important identified risk. Okay, now which molecule we should consider? Tell me the molecule name also. Any molecule name which could cause anemia? Tell me guys, any molecule name which you have processed in ICSR and the anemia was listed.
Clozapine induced hemolytic anemia.
Clozapine?
It
Okay.
causes anemia.
Clozapine. Sure. So now, when we come to potential mechanism, here we will write the potential mechanism. What do you guys understand? What potential mechanism should we write? Anyone from you? What is the potential mechanism here? By saying potential mechanism, what are we trying to say? Come on, guys.
Mechanism of action of that drug which causes anemia means like some.
Give me example. Give me example. What do you want to say? Don't worry. Just tell me. Nothing is right or wrong. We are here to learn.
Yeah, so if we are talking about potential mechanism, so we will be just talking about the mechanism basically. So in what mechanism this molecule is working. So we wanted to check with the articles and the facts, the peer review we are having. So basically, the potential mechanism deals with the biologically what how the medicine is working with the biologically and what acts it what's the mechanism of action of it. So we
Okay.
will be just checking it from the peer review and the sources we are having. From there, we will be checking the potential mechanism. So how does it
So,
work? Yeah.
so you mean to say that we have to look for the mechanism of action of clozapine here as to how it is working with the
Yes.
body,
Yes.
right? Okay.
Correct.
Any other thoughts? Any other thoughts, people? If I'm asking this question, that I usually say in previous classes also, that there must be some reason behind this, because people will tend to confuse you with this question and most of the candidates get confused.
We need to
So,
check the mechanism of action of the drug, whether it causes anemia or not. Like
so that is the means that is the cause you are trying to, like, you are saying that whether clozapine causes anemia or not, right? You have to look at that, whether it is causing clozapine is causing anemia or not. So that is the evidence that you're collecting, whether for the causality assessment. See, you people tried very good. Someone said drug mechanism, for someone say how drug causes anemia and all those things. So you people tried very good and you are close to the answer. But the actual answer, if someone asks you what potential mechanism you are asking, writing here, so you have to tell them that we look for the articles, open source, because this mechanism might be provided into your RSI. See, your RSI is the standard document for everything, for whatever information you are presenting in PBER. So you will first go to your RSI and check whether under anemia risk the mechanism is provided. So your RSI is the first source. Most of the times it won't be there in RSI. The mechanism are not there in RSI. So what becomes the next source here? Our source then becomes open literature. Okay. Source could be RSI. I am writing here. Okay. I will share this document again with you guys today. Open literature then. If the mechanism is not provided in RSI, then the source will be open literature. Now let's come to the question, what is the potential mechanism here? So by saying potential mechanism here, we mean that we have to look for the information how clozapine and the pathophysiology of anemia is related. Means there is certain pathophysiology of anemia. If it is a disease, there would be certain pathophysiology. There would be some mechanism it is going through to have that anemia, or certain changes in the body which makes us to have the anemia. Okay. And we will have a mechanism of action of clozapine, how clozapine is acting into the body. There is a pharmacodynamics as we have studied earlier. So clozapine has having its own mechanism of action. Anemia is having its own pathophysiology. So we try to check from the literature as to how clozapine's mechanism of action is hindering with anemia, or how is it related with the anemia's pathophysiology so that it could cause anemia. Did you get the answer or you are confused? People.
Please take them.
Okay. See, here what will happen. So let me think of an example. Suppose clozapine is quite typical in its mechanism of action. Uh, Let me think of some other example. Other example we can have, like we are using that uh, we have a well-known risk of ulcer, or you can say bleeding in the gastrointestinal region from the this what we call the NSAIDs which we are using, right? Are you people aware of that? NSAIDs or
Yes.
like ibuprofen and all, we have a risk of that ulcer or bleeding gastrointestinal tract, right? So, are you aware, right?
Yes, ma'am.
Okay.
Yes.
So what happens if we see ibuprofen and all, so all the NSAIDs, so they have a mechanism, means how they act into the body, they inhibit the prostaglandins, right? Prostaglandins in the body. So the same what happens in the gastrointestinal tract, the prostaglandins act as the you can say savior for the GI tract. Prostaglandins act as a soothing layer for the GI tract. So our paracetamol, or you can say ibuprofen, when it is inhibiting the action of prostaglandin, because prostaglandins are mainly responsible for causing pain, right? So they will inhibit the prostaglandin, then only their mechanism of action will be there and they will relieve the pain from your body for which you are taking that drug. Now, this inhibition of prostaglandin causes harm to the GI tract. So that soothing nature of prostaglandin is not able to execute into the body, due to which the prostaglandin inhibition, due to which we have a bleeding risk associated to this drug. Did you understand? They are inhibiting prostaglandin to relieve the pain. So that is their mechanism for which they have been marketed, or for which they have been approved into the market. But that inhibition of prostaglandin is leading to the risk of bleeding into the GI tract. So now you can see both the mechanism of action, how they are interrelated, why it is causing, why it is having a risk of bleeding. People, are you with me?
Yes, Amit.
Yes, sir. With this, you understand.
Yeah. So this is how we try to search the potential mechanism for any risk associated with that drug. Because this is the common, so we know it is most into the market from the 60-70 years, so we are aware of that. So in the same manner it happens, like in this example you people have given, anemia is an example of risk with the clozapine. So we will go and look into our RSI. If they have already provided into the RSI, then no problem, you can copy paste it from there and give the reference of RSI, no issues with that. But in the most of the cases, that it won't be there in RSI. So what you will have to do, you will have to go to the open literature, search for clozapine and anemia, get the literature, whatever you get. In that literature, you will have to look if the mechanism of action is provided. The same mechanism of action you have to copy paste here and give the reference of the literature from which you have taken that potential mechanism. Till now it is clear? Till now it is clear, people?
Yeah, but how do we search for the literature?
That is the next step. I'll come,
Can I-
don't worry. That is the next step. But till here you are clear with what is the potential mechanism, because this will be a definite question. If someone is asking about characterization of risk, they might ask you, 95% chances are there, they will ask you what is the potential mechanism that you give here. So are you clear from the example of potential mechanism? What potential mechanism we are providing here? We are not providing the potential mechanism of the clozapine. We are providing how anemia could be caused by clozapine. That is the potential mechanism that we are looking at here. So we are to looking for the pathway, how clozapine's mechanism of action, normal mechanism of action, could lead to anemias. Correct? And you can give an example and that would be clear to everybody. Chalo. Then now, shall I move forward?
Yes.
Okay. Now we will go to the section on how we are going to search. अरे ये वाला नीचे नहीं होगा क्या? Our drug is clozapine and our risk is anemia. Chalo, how can we search? So first we will write clozapine and anemia. Simple. Okay. Now we'll start getting the hits. So National Institute of Study. See, there is a 2-year follow-up study on this. Clozapine induced anemia. There is a case report on this. Metallurgical side effects, Science Direct. So you will have to see what peer journals are provided. You can't take it from Wikipedia or something like that. Okay. A case Journal of Korean has provided induced anemia and thrombocytosis. So chalo first start with this case report. This is from National Institute of Health. So probably it is from PubMed given here. So it is a peer reviewed journal we do. So here they have given abstract. In the abstract, we cannot find the mechanism. Chalo, let's see. Here they are saying full text articles. Let's open the full text article. Mhm. They have not provided the full text article. No problem. If you're working for a client, then you will have access to all those things. So you can means access the full text article and all. Chalo, we'll go to some other then. Science Direct, definitely it would not be open because it requires access. Oh, full article is there. Chalo, we'll see what information is given here. We will go to see if there is mechanism of no discussion. No, they have not given a detailed information. Not giving the mechanism. Chalo, we'll see for some other article. Are you people following me?
Yes.
It extract. Only abstracts are available. We don't have access to full. Let me see.
Can you check with the DOI link? DOI link.
Just a minute, I have opened this. First, let me check this. Oh my god, it is in Korean language. Leave it. Which link you are saying?
DOI, published online. 31st November 2015. Because
Where
there
is
is
this one?
yeah, complete and right.
Amit sir, sorry to disturb, but here
Yeah.
in this abstract, they are mentioning that it is might be due to immune reaction. So can't we take like from the single sentence? Obviously, we don't have
When the statement is like this and you are incorporating into your document, then you have to say that the exact potential mechanism is not known. However, it is reported this that it might be due to, which may be a result of an immune reaction.
Okay.
And you can cite the reference here that you took this example, means this is a statement from this article. But
Right.
you will have to write first that, because it is not giving a clear mechanism, right?
Mhm.
So
Right.
it happens that you don't get the mechanism because it is a very rare condition that is happening with anemia, clozapine. So you might not get the exact mechanism from the articles. So in that case, you can write there
Okay.
is no exact potential mechanism known about uh, anemia related to clozapine. However, it is reported as a result of an immune reaction, and then you can cite this article. So this DOI is not opening, right? But it is not opening. Chalo, we'll see some other example. Aplastic anemia. This is asking to request PDF. Can we see? See, these are giving that smoking causes upregulation of. So this also can be included with the same statement that no, means the mechanism is not exactly provided. However, one statement was there, and the second statement you can take from here. So these are the challenges when we are doing it from personal laptop. When we are doing it through the organization, then we have access to all these articles so that we can get all those. In case if we are not getting from open source, we will try to put it here also. Clozapine associated anemia. Let's search what result PubMed gives us. You people also keep on studying. Wow, I am moving slowly. It was the same article I think we opened last time. See what is PRCA? Pure red cell aplasia. So this is how we need to do different hit and trial from the article, from the literature. Anemia happens. Chalo, we took the example of anemia, na, that it is a very rare scenario with this. So now, since thrombocytopenia is being caused, we can see that there is thrombocytopenia. So let's see what is the mechanism of action we have, because we have to learn only why to search for the one which is not happening. No abstract available for this. See if we are able to find clozapine associated thrombocytopenia mechanism of action here. Article is visible, right? People, are you with me?
Yes.
Yes, yes.
Good. Let's see if we can find something here. Mechanism they have not provided for this also. No. Pancytopenia is a combination of thrombocytopenia, neutropenia, everything. So let's see whether something is provided here. These are the free articles mentioned there only. That's why I'm opening it. Is report discussion. See, can you see for neutropenia they have provided here? See, this is the mechanism of action for clozapine-induced neutropenia. Can you see here? Can you see here, people? Can you read it as to how it is?
Yes.
We were searching for clozapine-induced anemia first, then thrombocytopenia, but here we can find for neutropenia. So this is how the mechanism of action is provided. Means neutropenia being caused by clozapine. So what is the mechanism? This is the second mechanism. The first mechanism they has provided above. So this, can you see as to how the mechanism of action is presented in the literature? And you have to do a lot of research on this because we don't get direct mechanism of action. So this is why aggregate is typical, because we have to do a lot of literature search and everything to write certain things into our report. So now it is clear how we write the mechanism of action.
Yes.
And you will cite this article into your PBER. When you are writing the mechanism of action, after writing mechanism of action, like suppose if you copy paste from here and go to the section where we were presenting it, in this section, you will copy paste it, okay? Let's see. No, not allowed to copy paste. Not allowed to copy paste. No problem. So you will write the articles mechanism of action here, refine it. If there are certain, like you refine it and then cite that article. How we cite the article? So we will write the numbering in the reference section, and then citing is provided in the, when we open this, see, here there is cite. So you will cite it. So this is in the format of Vancouver style of referencing. And this is the only format that we present until here before PMID. So this is how you will cite into your reference section. So first you will find the mechanism, you will copy paste the mechanism from there, refine it to have a language which is compatible with the report. Means there should not be that the he was, she was, something, no, like that. Simple mechanism of action, you have to refine it in terms of statement there. Then put a reference number. Suppose it is the reference number 7. So you will put number 7, and in the reference section, you will copy paste the site, whatever I have highlighted currently. So you will copy paste this citation into your reference section. Okay. So this is how you will complete the potential mechanism of action. Clear, people, till here?
Yes.
Yes.
I mean, sir, in actual also we search on Google.
Yes, we search on Google, or the client says they have given access to you for PubMed. They give access to you for Embase and all peer reviewed articles. So you can search from them. So Google also you can start, you can go to PubMed, you will have access, you can open the full literature and see. So there when you are working for client. Google is not a crime to open in client database, okay?
Okay. Any search string is provided or what for this?
No. This for this you have to only look for from your search string.
Okay.
Okay, that is what your skill will work here. Everything you won't have the search string. You have to make different search strings. Like we started with anemia and clozapine, we didn't get. Then I wrote clozapine associated anemia. Then also we didn't have. Then I wrote clozapine associated anemia mechanism of action. Then also we didn't have. But finally we found for neutropenia. Since we are learning, so anything would work for us. So it was just to show you as to what is the process. How do we follow all these things? Okay. Chalo. We'll move to next topic now.
Yes.
Chalo then. Then comes the frequency. So what do you guys understand from frequency? Tell me. What is your understanding for frequency?
It is common or it is uncommon. How frequently that event is. How that risk is due to that drug.
Okay.
Number
What
of
else?
there with that
Number of.
cases reported with
Okay.
that.
Okay. What else? What else, people? And what could be the source for writing this section, frequency? From where we will get this information?
Clinical study reports, CSR,
Line
Mhm.
listing.
cumulative line listing.
Okay. Okay. So first thing.
RSA also there is.
Okay. So chalo. As you people told me that frequency is rare, very rare, common, very common. So that is provided in RSI. Okay. From there you will take, correct?
Yes.
So tell me one thing, like suppose if you have written a PBER last year and you are writing a PBER this year. and you are taking the frequency for from RSI as common, very common, rare, very, will there be any change in this section from the previous paper and the current paper which you are writing? If you are taking frequency as rare, very rare, common, very common from RSI,
No.
will there be any change? Any change? It will be same, right? Because RSI tells you what is the occurrence of means event. That is rare, very rare. I understand that is called as frequency, but for every paper it will be the same, right? There is no change in frequency. So what's the use of writing this section then? So now, always remember that frequency is about the number of events that happened with your drug. That is the frequency here that we are talking about. It could be from the clinical trials. It could be from the post-marketing cases. Okay. Did you get my point? The number of events that happened.
Yes.
Okay. And now from clinical trials, what happened? We get mostly the serious adverse events that are reported from the investigator, right?
Amit, I have one question. Number of
Yes.
total number of events happen or any specified number of events, specified number?
You are writing about anemia here, right?
Okay. Understood.
So, yeah. So total number of anemia cases that were reported. Okay.
Okay.
Chalo. Then, yeah. Yes, tell me. Somebody was asking. No? Okay. So let's move. So what we considered here as to how many times anemia cases were reported. So there could be two sources, ideally. One is from the clinical trials that happened for clozapine, and the other is when it is into the post-marketing phase. So whatever ICSR cases are being reported, correct? Correct, people?
Yes.
Yes.
Okay. So from clinical trials, how will you get the information?
Yeah, if we go in the in the E3, like clinical study report, the CSR report, from the day when the clinical trial approved for that now. So from there, we got the whole details and everything in clinical study. We will go and just check it the frequency means from there on.
Okay. Okay. So CSR could be written for which clinical trial? Do you have any idea? Means, what are the CSR? Do you people have idea of CSR first? Clinical study report, CSR. Any idea? Anybody has any idea? Anybody? Because this is the first basic question somebody will ask you, now, like what is the CSR? When you are explaining them that you are taking information from CSR. So see what happens.
Can I
Yeah,
tell
tell me.
something?
Yeah.
Amit, a clinical study report is actually it's a detailed documentation part where we can just summarize the whole results and data of clinical trial. So till now, what are the important comprehensive overview was there with the in the trials methodology. Till now, we have received upon conducting the clinical trial process. So in this section, basically, there are lots of like it's a in the E3 part now. CSR is an E3 part. So in the E3 part, we will get the whole details, like from the introduction section to study
So
design.
that is you are talking about the CSR format. Don't go to E3 because that is the guideline that is provided how to write the CSR. Like for PBER, what is the guideline we use to write the PBER?
E to C
Any- E to
R3.
R3, right? In the same manner is for CSR. So if somebody asks you what is CSR, just tell them, okay, you are talking about clinical trial. So which clinical trial we are presenting in the CSR? Which clinical trial? Each and every question I am asking so that everything is clear to you people, everybody who is on the call. So which clinical trial, for which clinical trial we are preparing the CSR? Any answer? Which clinical trial we are preparing the CSR? People, which clinical trial we are preparing the CSR? No answer. Okay, chalo. Suppose you are investigating clozapine for the disease schizophrenia, suppose, right? So you would have some clinical trial or clinical study ongoing for that. There is some clinical study that is ongoing for that, based on which you have got the approval. Clozapine and schizophrenia. Schizophrenia is your disease in which clozapine will be used in the patients. So there would be one clinical trial, at least one clinical trial that was conducted to see whether it is if efficacious for schizophrenia, whether it is safe to be used in those patients. There would be some clinical trial conducted for that. Are you with me?
Yes.
Yes.
So that clinical trials information goes into CSR. This is a report. CSR is a clinical study report. The same manner you are presenting this PBR, you are writing this PBR for a molecule. So CSR is a report for that clinical study, that clozapine used in the treatment of schizophrenia, that clinical trial which was conducted for that. So for that clinical trial, a CSR, a clinical study report is prepared, in which you will tell all the information, that why this study was conducted, what was the method that was taken, what was the method to analyze the results, how the safety information emerged, what was the safety information emerged into this section, then what is your efficacy information that emerged into this clinical trial, everything, and what is your conclusion and all. So it is basically a report which explains about that clinical trial which was conducted for clozapine and schizophrenia. So now you are clear what is CSR?
Yes, Amit.
Okay. And somebody would ask you as to when do you consider, because this is a common question. Somebody would ask you as to when you consider CSR as complete. Well, what happens, like sometimes people say, like clinical trial, your clinical trial team in the same company when you are working, you will face this situation that your clinical trial team will say that the study is completed, because they say when they say that the study is completed, when the last patient last visit is done and the study is closed, means they are not doing anything on those studies. The study is closed. The last patient last visit is done. Now there is no clinical trial involvement in that clinical study, clinical trial team involvement in that clinical study. So they will say that the study is closed out. From their perspective, last patient last visit is the clinical trial closed. But from regulatory perspective and from our perspective, who are writers for the reports, our perspective, that CSR is completed when, or the clinical study you can say is completed when the CSR is signed by the authority, whosoever is responsible for signing the CSR. When he has signed and approved the CSR, stamped with the date, then it is called as approved CSR, okay, or completed CSR. Then only we can take information from that CSR. So when is the CSR considered completed? When it is time stamped and approved by the concerned person or authority. Am I clear? When CSR is completed?
Yes.
Yes.
This will be a question people will ask you when you consider the CSR as completed. Okay? So don't confuse. Chalo. Then we will come to this section, frequency. So in CSR, you will see, if you have, you can find CSRs available on this open source. So in CSRs, you will see that there is a safety section for those CSRs. In those safety sections, there will be the serious adverse events that are reported in that. So in clinical trials, what happened now, they will provide a table, like they will provide a table that there is a control group, there is a placebo group, there is a treatment group. Treatment group could be one or two. So from table wise, they will present the information, like anemia, if they are discussing. So they will tell that anemia was in how many patients it happened. It might be in percentage, it might be in numbers. So it might be in both. So from that table, you have to take the information like anemia was, and the same has to be pasted here, that anemia frequency in this treatment group was this much, in placebo group was this much, in in the control group was this much. So you have to give that same table of anemia into this frequency section. Okay.
Yes.
Am I clear?
Amit, can you just tell us that in which section in the CSR report this table is there? Like, do we have to-
They have given a serious adverse event section is there, or safety specification section is there. You can find from there. I don't remember the section name number, but there is a section on safety part. So you can find from there.
Okay, okay.
Okay. So there is no specific section like means the number I don't remember what is the number, but we go to safety section and go to the anemia risk wherever anemia is provided. What its frequency is provided for anemia, we take those frequency and paste it here because that is from clinical trials and we are not means accountable for that. We are accountable to take the correct information from that CSR. Okay. So did you get my point as to how anemia frequency is provided from clinical trials? You will not go to open source. You will go to your CSR. Remember always. Okay. CSR, I will write the source. I am writing the source here. Don't confuse
I
anything.
have a question.
Yeah.
Will there be any instance that a CSR is not available for any study?
Yes,
how
there will be a SAN. So
we will
in that case, what you will do for clinical trials, you will write like this, just for an example, clinical trials, we provide the frequency like this. So for clinical trials, you will write not applicable. Okay. Yeah, not available. Simple. For clinical trials. Okay. There will be a section for post-marketing also. In this way, we present the frequency. So did you get, if CSR is not available from your company, you won't go to open source and provide it. You will just write that not available. It happens in generic cases, generic molecules. Okay.
So, Amit, can you?
Your voice is
Hello.
not clear. Yeah.
Yeah. So I was asking, like, let's say for a molecule, so 2 to 3 clinical studies are being done at the same time. So we have to refer all the CSR.
Yes, all the CSR for anemia. In all the CSRs, you will look if information is provided for anemia and whatever frequency of anemia is provided, you will provide there. Mentioning the study name. Like suppose one study is a study C234, another study is a study D365, another study is E567. So you will write study C234 first and provide the frequency, then you will write study D234 and write the frequency whatever is provided, then study third and the frequency whatever is provided. Okay.
Oh, okay.
Clear how to populate frequency from clinical trials? Let's
Can
move to
we
post one.
see
Yeah.
any example like we saw for potential mechanism for this also? Frequency, if possible.
Mechanism, we will have to look for CSR then.
Yeah, Amit, can we get any CSR report so that we can just have some idea of it?
You will have to search the open source. Because I can't share any CSR from my company, right? So we
Amit,
have.
actually, I have downloaded one CSR. Can I that means I'm also in a condition that I need to understand that means how to get the data from this. Can I share the screen?
Share it. Here's the chat. Have you shared?
Option. There is an option to give the control or because I'm not able to find any option.
Can you share the file with me?
Yes.
On chat. Hmm.
This is a random CSR I have.
No problem. Let's check.
I have shared.
But I'm not seeing here.
Are you able to see my screen?
No. Anybody else can see the screen?
Yes.
Yes.
Okay. Why am I not able to see?
Yes, we can see.
Why I cannot see? Yes, I can see now. Sure. Mera, I will stop share. Zoom it a bit. Open the bookmark section if you can. This is a non-interventional study. This is not clinical study. See the topic heading, a non-interventional prospective open label study.
Actually, it's showing some CSR related.
CSR might be there for non-interventional study, no problem. But it is a non-interventional. It is not a clinical study.
Okay, okay.
Okay. So we will check what CSR is available on open source, and then maybe we can see for this frequency information in next class. Okay. Because if I'll start searching and then it will take time again as we were doing for mechanism of action. So it's fine now if we look for the CSR. You people also look for the CSR information, the clinical trial, and then we will see as to how we pick the frequency from that. Correct? Guys? Is it fine?
Yes, yes.
Okay. But this table will be there. Don't worry. The table is there exactly. You have to copy paste from that. So just you will have to have an idea. So we will look for the CSR, what is available open for open source, and then I'll let you know. Okay. Otherwise, chalo, ठीक है. We'll let you know once the recording is off. I'll tell you one thing more. Chalo. Clinical trials we are done. So how we take the frequency for post marketing? Okay. So always remember one thing, that from 16.4 onwards, we represent cumulative data into the PBER. Before 16.4, we are presenting mostly interval data. But from 16.4 onwards, we present cumulative data. What is the difference between interval and cumulative? Anyone? What is the difference between interval and cumulative?
Interval means the perfect. Means, for example, we are taking from January 23 to 24, and cumulative means from first DIBD. From that time, we are taking all the data from start to end.
Correct. So always see, always say that interval is the reporting period of the PBER. Okay. Like then you can say, for example, if the reporting period is from 1st January 24 to 31st December. You have to mention that reporting period as a term. That is how the people catches whether people are working or not. The other candidate is working in aggregate or not. So we are frequently used to writing this reporting period, interval, cumulative. So you have to use the term reporting period of the PBER. And cumulative is from the date of inception of the molecule into the market. It might happen that suppose clozapine is there. So you are an MAH for clozapine. There is another MAH for clozapine. Their IBD was something else from previous to you. Like suppose your IBD is 1st January 2010, but clozapine came into the market on 1st January 1990. So you have to, however, the cases will be there in your database from 2010, but you will pull the cases from, like we used to do, we pull the initial data as 1st January 1900. And from that, till the DLP of this molecule. Like in this case, if we are saying that 31st December 2024 is our DLP, then 1st January 1900 to 31st December 2024. So we will pull a line listing for that will be called as cumulative. Okay. It is not necessary that you have to start from your IBD only. It can be from 1st January 1900 to till date, the DLP of that day molecule report. Okay. Okay, clear? Cumulative information.
Yes.
Am I clear?
Yes, Amit.
Okay. So 16.4 onwards, we use cumulative.
Sorry to interrupt, Amit. Can you again repeat about cumulative?
Cumulative is ideally, if someone asks you cumulative, then you can say that since the IBD or the first approval date of molecule for us till the DLP of this report. Suppose example, the reporting period for your PBR is 1st January 2024 to 31st December 2024. So in this case, what is your DLP?
1st January.
No, DLP is your 31st December 2024, the last date. Okay. The data lock point, 31st December 2024. That is your DLP. Okay. Now suppose this clozapine molecule is first approved for your company on 1st January 2010. Okay. So that is the first approval date for your molecule. Correct? So from 1st January 2010 to DLP 31st December 2024, you will pull the information that is line listing that is called as cumulative line listing. First thing, correct? Now the clients or the companies, what do they do conservatively? Initial date which you are putting here as 1st January 2010, they put it as 1st January to 1900. And the DLP will be 31st December 20. DLP will be fixed. It will not be changed. 31st December 2024. So they will pull the line listing. Whatever cases are there, it will pull up and you will get the cumulative line listing. Correct? Clear?
So 1900.
1st January 1900. Safer side. That is not a means thumb rule. See what will happen. 1st January 2010, your drug was approved first time. So your database will have information from 1st January 2010 till current date. Are you getting my point? When will you start processing the cases in your database? From 1st January 2010 only, you will start receiving the cases and you will process it into your database being a MAH, correct?
Correct.
Are you getting now?
Yes.
But
why 1900?
1900 because it might have happened that clozapine was approved on 1st January 1900. Suppose 1990. Some 10-20 years ago. So safer side they have kept as 1st January 1900. It is not necessary if you answer that from my date of approval for post-marketing cases. From your date of approval, that is 1st January 2010 in this case. From that date till 31st December 2024, you will pull the line listing that is called as cumulative line listing. Correct? Don't confuse with 1900. Leave that concept. Okay?
Okay.
Now it is clear the first date when you got the approval and then. Okay. So that 1900 concept is also there because you could have, suppose if you are an innovator, then you could have conducted a clinical trial. So you will go for DIBD then. What was the DIBD for that molecule? Because clinical trial cases will also be documented in your clinical safety database, correct? So from the DIBD, you can take this. So that's why we keep it 1st January 1900 as safer side so that all the information is captured into your cumulative, whatever cases are there in your database. Now,
and if we know the IBD for other companies before our IBD, then
IBD for other companies is not required for you. For safer side, you can keep 1st January 1900 and till your DLP, you can pull the information. No need to worry about others IBD, what is there. So that's why we keep 1st January 1900 as a start date and 31st December 2024 your DLP as a end date for that line listing. Confused or clear?
Clear.
Okay. So we will pull the line listing for that cumulative for getting the post-marketing cases. Okay. And the search criteria will be the same that you have used to pull the interval line listing for section 16.3. Do you remember? Do you remember, guys? People, do you remember the search criteria that we use for the risk in 16.3? Yes or no? No. Hello?
Yes. Yes,
You,
yes. Yeah.
I think you are forgetting. So at the time of KOM itself, you have decided which risk to be taken, what would be the search criteria for that risk, correct?
Yes, Amit.
Which was endorsed by physician. So same here, the same search criteria of anemia, which you have decided earlier, the same search criteria will be used, only the reporting period will change from reporting period to cumulative reporting period. So it will be from 1st January 1900 to 31st December 2024 in this example. Okay, and you will get the line listing for anemia cases, correct? Now, when you get that line listing, you don't have to do anything. You just have to look as to how many cases are present in that line listing. Suppose in reporting interval it was 50, but in cumulative we found it to be 5,000. So you will have to write here that a total of 5,000 case reports were reported from post-marketing or global safety database related to anemia. That is the frequency of anemia risk in post-marketing. Did you get it?
Yes, Amit.
Okay. So we have completed the clinical trial and post-marketing frequency, how we are presenting.
Amit, we also have to write that how many fatal case we have received in
We are coming to that. The next section
likewise
is there,
also,
na?
no?
Seriousness or outcome.
Okay. Yeah.
There we will write about all those things. So here just we write the numbers from clinical trials also and from post-marketing also, because that is frequency only.
Okay.
Okay. So what is your source from here now? One CSR and the other is global safety database. That is either Argus, RSG, Scepter, Sapphire, whatever you are using for your company. Okay.
Okay.
Source clear for frequency?
Yes, Amit.
Sure. Now let's move to seriousness or outcome. So for clinical trial cases, means clinical trial seriousness information. So it is clinical trial information is pretty easy because you have to copy paste from CSR. So in the same section, when you will go after the frequency, there will be seriousness or outcome provided. How many were recovered, how many were not recovered, how many turned into fatal, how many were life threatening. So all that things will be provided. All that information will be provided into your CSR in anemia section for that, where anemia information is presented. So like the frequency table is there, the same way you will have the seriousness or outcome table in that. So outcome table will be the outcome not recovered, not recovered, whatever outcome that you do in ICSR, na, the same way it will be there. And there would be that seriousness, for seriousness, there will be a table in CSR, it is named as severity and nature of risk, or you can say severity of risk from that. Severity, by severity, you understand, you would have heard about the term grade 1, grade 2, grade 3, grade 4 adverse events.
Yes.
Grading is there, grade 1, grade 2, grade 3, grade 4. Grade 4 is most severe, grade 3 is lesser than that, than grade 2, grade 1.
Yes.
So in CSR, you will find severity to seriousness as grade 1, grade 2, grade 3, grade 4. Means anemia is how many grade 1 cases were reported, how many grade 2 cases were reported in clinical trials, how many grade 3 cases were there, how many grade 4 cases were there. So this type of severity will be provided as a seriousness term for from CSR. Did you get it what I am saying? People?
Yes, Amit.
So by seriousness from clinical trial, we mean grade 1, grade 2, grade 3, grade 4 seriousness. Okay.
Yes.
And from outcome, we mean the same outcome as in ICSR, recovered, recovering, fatal, life threatening, or not recovered, recovering with sequel. The same outcome is there as you present in outcome. For clinical trials also, it will be in the same manner. Okay. Now, from global safety database means for post-marketing, how we'll get this information. So there is a column of seriousness and there is a column of outcome in the line listing. You have seen the line listing last time?
Yes,
in the same way, there will be a column of seriousness and line listing. So when you will go to your line listing, cumulative line listing that you have downloaded, put a filter of seriousness. So you will put filter of seriousness as serious. So you will get these many cases as serious. Non-serious, these many cases are non-serious. Then in outcome, you will see fatal first. How many fatal? Then life-threatening. Then how many other serious? Means hospitalization and all. And then no need to provide non-serious information. Only serious information we provide you. So this is how you will provide, and then outcome and seriousness for post-marketing is also done. So this is the way you provide seriousness and outcome for clinical trial and post-marketing cases. Am I clear?
Yes.
Any doubt till here, people?
No, sir.
No doubt, right? So then move to risk groups or risk factors, right? So what could be the source for this? Source is always important for all of this. This is the only thing that is asked in the interview. First, how do you characterize? Then when you will explain them the parameters, they will ask what is the source. Tell me, what is the source for this risk groups or risk factors? What do you think? What could be the source?
RSI.
Correct. What else could be there?
Source kit, open literatures.
Open literature could also be there. Chalo. Mainly, the source is your RSI, because in RSI, most of the times it would be mentioned as to which patient characteristic is most susceptible to this anemia risk. Like for anemia, it would be mentioned that the patient who are having hemoglobin count lesser than 13 or 10 don't administer the drug. So the patient who are having hemoglobin count lesser than 10, they are the risk groups or risk factors for this. In the same way, it might be mentioned that the patient who are allergy means who are having a medical history of anemia. So this kind of thing would be mentioned in their RSI. So these are the risk groups. By risk groups, you understand the people or the patient who are before taking the medicine who are prone to having this anemia risk. So that could be mentioned in your RSI. Did you understand the concept? What is the risk group and risk factor for anemia or any disease or any risk, you can say? Are you with me, people? Yes, no, maybe.
Yes.
Yes, yes.
Okay. So most of the times you will find it in RSI. If it is not there in RSI, then you can go to open literature again and search in the same manner, like risk factors for anemia, risk group patients for anemia risk factors, anemia associated means patients who are more prone to anemia. This kind of search strings you can use and pull the information from open literature only when the information is not provided in your RSI. If this is not in your RSI, then you can go to the open literature, search for the risk group and risk factor, and in the same manner as you did in potential mechanism, take the information from that article, refer the article here, put the article citation in references section. Am I clear?
Yes.
Clear, people? We are
Yes.
clear till here, right? Chalo. Then we'll move to source or this preventability or reversibility. If something is not clear, do let me know. Huh? Source. What could be the source here? People, you have to tell me what could be the source here.
RMP.
RSI.
RMP is always there, because if your risk is there in RMP, then you will copy paste. Then no need to think about it. Forget RMP for current time. We are learning how to characterize the risk when there is no RMP. When we cannot copy paste from anything, we have to search and do that. So what could be the source in this case? Tell me people source. Initially itself, I told you that RSI is our main source for anything. So RSI definitely will be your source. Okay. So, but now tell me what information from RSI you will take. What do you think? What could be the preventability or reversibility information in RSI?
Routine risk.
From RSI, like we have warning and precaution section.
Hmm.
So like do not take if you have that.
Hmm. Hmm.
And
So your risk will also be in the warning and precaution section, correct?
Yes, yes.
So what could be the, you are going correctly, but think a little bit more. What could be the preventability or reversibility?
Contraindication.
Contraindication, first of all, see, always remember one thing. This is the mistake that you guys could do. Anemia is a risk. Now you are writing preventability and reversibility for anemia, not for anything else. Contraindication might be for hypersensitivity and all. So first, always remember that in RSI, when you go to the respective anemia section and not to any other section. So in this case, anemia, if it is an important identified risk, it will be in section 4.4, warning and precautions. Correct? Am I correct? Are you getting what I'm trying to tell you?
Yes,
Yes.
yes.
So in that anemia section, you will go and see what is the routine risk minimization provided by for that anemia. We discussed the concept of routine risk minimization and additional risk minimization, right?
Yes, yes, yes.
So there will be a routine risk minimization statement in the RSI under anemia's risk. Like they would say that monitor the patients who are having, monitor the patients or do the blood test for the patients and check if the patient is having hemoglobin lesser than 10. If it is having, don't administer the drug. This is the preventability. There would be some other criteria, like there is a liver case. So drug-induced liver injury is a risk. So in that case, they will write that check the AST, ALT or liver parameters for liver enzyme parameters for the patient. If he is having AST greater than equal to 3, if he is having ALT greater than equal to 2, if he is having SGPT, SGOT less than or greater than this, don't administer. So these are the routine risk minimization measures that are provided in your RSI respective to that risk only, which will come as a routine risk minimization here. Did you get my point?
yes, Amit.
Yes.
Am I clear?
Yes, Amit.
Okay, great. So after this class, go to the RSI, check what routine risk minimization is provided for the molecules which you are looking at. Any RSI you can download. I told you how to download SMPC from EPAR website. Okay. So any RSI for the current molecule you are working in ICSR also, go to that molecule, download its SMPC from open source, study that RSI as to which information we are talking about. We talked about seriousness, how to this risk group risk factors take from RSI. We talk about potential mechanism if it is provided for the risk there. Whatever we have talked in this section, whatever information you can take from RSI, download one RSI, try to look from the RSI as to what information I told you, is it available there, and then you can practice as to how we can present that information into this section. Okay.
Yeah, Amit, I have one question. Like,
Hmm.
this preventability and the reversibility now, basically for which section we will be mostly targeting, like in section 4.8, 4.2, 4.3, which we have discussed. Like,
I
so,
told you that
Mhm.
anemia, suppose if it is an important identified risk, then it would be under section 0.4 for sure. Confirm, right?
Yes.
So in section 4.4, you will go and see the subheading of anemia. Anemia would be provided there. And there will be a paragraph or two paragraph on anemia. In that paragraph, you will look what is the routine risk minimization provided. And the same you will copy paste here.
Okay. Okay. Yeah.
Okay.
Yes. Yes. Okay. So we will just check with the routine risk minimization measures, where we will be finding the which is the routine risk we will be finding one. After that, we will go into the other side. We'll check whether it is there or not, and that time we will just go and copy that data and we will paste it right in the paper report.
If routine risk minimization means if it is a risk, important, identified, or potential, the routine risk minimization would be provided mostly in that from there you can take it and copy paste here. Reversibility is something different. Like it is not routine risk minimization, you can say it can be a routine risk minimization, but what happens now? Like suppose in the RSI it will be provided, suppose the patient is having hypersensitivity reaction as a important identified. So when you will go to hypersensitivity section in sections 4.4, under that you can see that it will be provided that if a person experiences hypersensitivity reaction, immediately stop the drug. So that is a reversibility, right?
Yes, yes.
And also a routine risk minimization, preventability also. So preventability mainly what happens that before happening, that means before starting the drug, whatever you measures you are taking so that the patient doesn't have this risk, that is called as preventability. But once the risk happens, occurs with the patient or the event occurred with the patient, then what measure you take to reverse that risk or the event? In this case, as I mentioned for hypersensitivity, stop the drug immediately. So that is a reversibility. So you can write it here, the same statement. So now did you get the difference between preventability and reversibility?
Yes, understood.
Preventability is a preventive measure before starting the drug itself, and reversibility is once the event happened, then what measure you take. Chalo. Any other question? Chalo. Then we'll move to impact on individual or public health. So what do you understand? What could be the impact? How can you say that it is an impact on public health? Like suppose if a patient is having some event, he had certain course of actions during that. So how can you say that it is having a public health impact? What could be the parameter that could tell you that, okay, this is the public health impact?
Any fatal outcome?
Correct.
Uh, Actually, I think it's a severity. Um, and specifically any population which is getting affected, we can check with that. Then we'll call as a risk. So these are the public health impacts, like the broadly, like for the specific type of like ASEAN or like Caucasian people are very affecting on that. So it's a population affected. So we can call it as a public health impact in it. And for the and suppose any specific significance of that public health impacts if affecting in like in terms of morbidity mortality in future, that impacts the quality of life as per my understanding actually. So this would be there as an measure the public health impact. So
Okay.
at that time we can consider as a risk. Yeah.
Okay. So fatal, life-threatening, and all these are is definitely a public health impact if it is having, because it is a most severe form. Now, in most of the risk, we don't find that much fatality and all those things. So what happens? Just for your understanding, I'll tell you in layman language. Try to understand. Like suppose if you had anemia, you are a patient, single patient, you had a anemia. So will it impact your daily activities? Can you work having anemia and have medicines and you can work and all those things? Do you have any
Yes.
impact on anemia when you are having anemia? Do you have any impact that you cannot do your work, day-to-day activities that you are doing?
No, it's
Yeah,
not.
due to dizziness or something.
Means suppose, have you seen the people who are anemic and working?
Yes.
Yes, we have
Right.
seen it.
Suppose if you are having a cold or flu, or suppose if cold is your risk, when you are having a cold, can you work or not? Is there any impact on your daily day-to-day activities?
Yes,
Will you
there
be
is.
will you be hospitalized for cold?
No.
No.
See, if you have any disease, like suppose if you are having a pain also, then also you will think that I can't work. But you can manage your, there is no impact on your day-to-day activities, right? You are not hospitalized for that. You don't have to be under observation of doctor to monitor that, right? Whether it is anemia, whether it is cold, whether it is some fatty liver or something kind of things. So these are all the parameters in layman language, if you understand, that you are not getting hospitalized for it. your day-to-day activities are not being hampered, then that is not a impact on public health or individual patient health, you can say. Okay. Like anemia. Anemia, in this case, you won't be hospitalized. It can be managed through OPDs also. You can get the medicines, you can keep on taking the medicines, you can continue with your day-to-day activities. So this is not impacting the public health or your individual day-to-day activities. That's why there is no impact on public. Did you
So
get
it
the
is
concept?
linked to severity, right?
Yeah, it is linked to severity, means how it hampers your day-to-day activities. After having that risk, will you be hospitalized immediately? Or it can be monitored through OPD settings or clinical or ambulatory settings, you can say. If something hampered your day-to-day activities, means you are hospitalized or you are means your doctor advised you to be on complete bed rest, you can't do anything. So these kind of risk or events are tend to have impact on public health, because if it has happened, it will hamper your day-to-day activities. But anemia, flu, as I gave you example of other fatty livers, hypertension is also not having a public health impact, because you can work having hypertension, cholesterol, diabetes. So these are not impacting the your day-to-day activities. Okay, so there is no impact on this, right?
Yes, Amit.
And the same here, you won't have that information into your RSI. In RSI, what could you take from RSI? Like suppose if it is a means there would be the routine risk minimization that is provided, that you can paste here, or simply you can write that this disease, like suppose anemia we are talking. So we can write something about that anemia is a very common ADR with the molecule treatment, you can say, and then you can refer to section 4.8 in which you have presented anemia as very common or something like that. You are saying very common, so it is a frequency, so it would be supported based on the RSI. In RSI, anemia will also be very common in 4.8. So you can refer that, and you can say that uh, uh, since it is very common, it can be managed in the clinical or ambulatory setting. It is not considered to be having a public health impact. And you can code this. Okay, so you can code the uh, RSI frequency from section 4.8. RSI I'm talking. And if any relevant routine risk minimization is provided. So now did you get what is called as impact on public health or impact on individual health?
Yes, Amit.
Okay. So now let's come to the section of evidence source or strength. It is nothing but like you, what source you took. Like you took CSR, so it will be your source. You took RSI, so you will mention the RSI name as to SMPC dated and so and so. Then you took open literature, so you will write literature simply here. Okay. And global safety database. So these were your sources. Whatever sources you use for above sections, you will write here as evidence source or strength. This completes our characterization of risk. Correct? People? Any doubt till here?
Amit,
What
apart
is
from
the
these mechanisms, these are the categorization, na? So the categorization of,
Categorization nahi, characterization.
sorry, sir,
Always
sorry.
use right term, otherwise other person will catch you.
Yeah, these are the characterization of risk. So apart from this characterization, so any other, like apart from this, any other parameters we are having?
No.
Okay, okay, noted.
Sir,
so
is
Yes, tell me one by one.
Hello, Amit sir. I have seen one paper that one paper we are given of COVID-19 that AstraZeneca molecule. So in that it is written as means impact of on risk benefit ratio in these parameters.
Risk benefit ratio is simply the public health impact only. No need to write that. You can leave that.
Okay.
Don't write about risk benefit. That is basically a topic of
Amit,
RMP. Yeah.
yeah, so this characterization, what is the objective of 16.4? What are we trying to say? After characterization, what are we trying to
We are justifying as to why we have considered it as important identified, why we have considered it as important potential. So based on these parameters only, we have done that categorization. So this characterization gives you a evidence of its categorization into important identified or important potential.
Sir, for every other we have to write different, right? For important potential, we have to write different. For missing
No,
information.
no, nothing different is there. The same parameters will be used, the same procedure will be used as I told you, just the name of the risk will be different and the articles and every other things what you have searched will be as per that risk. Like suppose here anemia was important identified risk. Now suppose drug induced liver injury is important potential risk for clozapine. So whatever parameters we discussed here, like potential mechanism, you will see how clozapine could cause drug induced liver injury. What is the frequency of drug induced liver injury in clinical trial and post marketing? What is the serious and outcome of the drug induced liver injury reported events in the CSR and safety database? What are the risk factors for drug induced liver injury to happen? Drug induced liver, who are those patients? Preventability for that. So you will write from that perspective. The risk changes, the perspective changes. But there is nothing like in important potential risk when in 16.3 you were doing. So for important potential risk, you have to look for causality assessment. But for important identified risk, you look for severity and frequency. So that is not there. The parameters will be same. The approach will be same. Just the name of the risk will change.
Okay.
So this is for important identified and important potential. I'm not talking about missing. Missing, I'll let you know. Just one more thing. It
And
is a
for
very
every other risk, we have to mention different means one by one, right? For every risk.
You only think, tell me, will the potential mechanism be same for anemia and drug-induced liver injury with
No,
clozapine?
it will be different.
So definitely you have to write different, na?
Yes.
Mr. That means we are going to mention this characterization in the tabular format only while preparing the paper, isn't it?
Tabular format most of the companies prepare, but you can write, means you will have to make a separate table then for each thing. Like suppose tabular format is only preferred. Like suppose if I have mentioned it like this, one column for parameters and one column for whatever in observation we got. Okay. The second format could be you can put potential mechanism here and below that only you can write the information, this details information. But it has to be in tabular format because it gives good presentation. That the same way you can write here, no need to have this second column. You can delete this second column and write below that. That is the only thing that you can do. But it is always provided in tabular format only because it is easy.
Sources must also be that means mentioned somewhere.
What is?
Sources should also be mentioned somewhere, or that means
Sources
not.
I told you na, like suppose if RSI is source, then you have to because your RSI will be in the reference section. If you go, your RSI will be on number 1. So you can write number 1. So if you have used some literature, then you can write the numbering of literature and provide it into the reference section. So source you have to mention the source as a reference in this table. Because that is the basis on which you have provided the information. This is a scientific document that you are writing, right? So you should have reference for whatever you are saying.
Okay, and are we going to summarize this characterization of risk anywhere? And would it be going to that means use in any type of such reporting or something like that?
We are summarizing only. We write in crisp summary in this section, and this information you can find in RMP also. It is used for RMP also. Wherever the risk information is there, it will be presented there as a characterization.
Okay. Okay, thank you.
It is in summary format only. Don't worry about whatever you are writing here. It will be crisp summarized form. It will not be a whole literature. You will not paste. You will paste the relevant information from there, right? So it will be in the summary format only. Like you only shared that Vaxzevria fever, so you can see there it is provided in summary format only. But that is a vaccine that has provided lot of information. You can look for some drug. So it will be easy.
Okay.
So, in that paper for COVID-19 vaccine, I have seen that
Voice is not.
RSI was.
Your question is not clear.
Hello, can you hear me?
Yeah.
Yeah, so I was saying that in that paper which you have shared, COVID-19 wala. So I have seen that they have mentioned that RSI for this paper will be the updated RSI, and you said that no, whatever was effective on the first date, we will take it as a source. So which it will be?
The RSI which was effective at the start of the reporting period is your main RSI. Okay.
Yes, but that section, section 3, I think, where we have to mention the changes in RSI, section 4, section 4. So where they have mentioned that the source for this paper is updated whenever they updated the RSI in that reporting period. So that updated version, they are taking it as a source.
So this is not a thumb rule. For some companies tend to do that whatever RSI was updated during the reporting period, they will provide that RSI. They will use that RSI as a updated information. But what happens, like suppose most of the companies, what do? They will keep the RSI which was effective at the start of the reporting period. The reason, I'll tell you what is the reason behind that. Suppose there is an RSI version 1 at the start of the reporting period. Okay. Then the RSI version 2 became effective during the after this means let's start with the reporting. 1st January 2024 to 31st December 2024 is your reporting period. On 1st January 2024 means the start of the reporting period, RSI version 1 was effective. But in June 2024, there was another RSI updated at that time. So now what happens from 1st January 2024 to January 2020, June 2024, whatever ICSR cases were processed on which RSI basis their listed was done. Can you tell me? The listedness of the cases from 1st January 2024 to June 2024 was done on basis of which RSI? As per your understanding? First one.
Which was effective on 1st
Correct.
January.
Correct. And from June 2024 to December 2024, the listedness was done on version 2. Right? So if you are taking version 2 as your reference RSI, then the listedness would be wrong from 1st January to June 2024 for the cases. Correct? But if you take listedness based on RSI version 1, then your listedness information is not incorrect for the whole reporting period. Correct?
But it might change in the second version, right?
That's what I am coming to. But it won't be incorrect, right? It might change in second version, but it won't be incorrect whatever you have presented. Means you are not giving some false information, right? Now what happens when you say that your RSI is updated? So when in aggregate you are analyzing the cases, you will use you will see both the RSIs. What is the safety difference in RSI version 1 and version 2? Because then you can find that what were the safety, you can say the adverse events or the newly added ADRs or all those things. Those you can find from section from the version 2. What if there is some ADR added? Ideally, what happens when a RSI changes? There is no such like ADR is added. There is some statement that is changed into a risk or something like that. It might happen that sometimes ADR is also added. So you can find from there would be a difference between the RSI version 1 and version 2. There is an annotated copy available. So you can see from there that what ADR was added into the section 2, version 2, or what was deleted in version 2. So that you can, while you are writing the aggregate report, you can say that this was listed as per this was unlisted as per version 1, but in version 2 it was listed. Hence, for this aggregate report, it is considered as listed. So in this way, you can write your statement. So that's why we use the RSI, which was effective at the start of the reporting period. But we present the information in section 4 if it is updated. What was the updated safety information in that? There is a section. You remember, na, when we present that the RSI was updated. So we have to present also the safety related information what was updated, correct? So from there you have the information as to new safety information. What is the new safety information in version 2, which you can write down in the your analysis part when you are writing. Am I clear? Why we use the RSI at the start of the reporting period? Still confused? Hello?
Can you hear me? Hello?
Yeah, tell me.
Yeah, so that is clear. This question regarding characterization of risk. So as we were capturing the risk from RSI, so we were looking only for frequency, severity, causality, all this. Apart from that, we were not characterizing fully like
I'm not getting your question. Why do we look for frequency from RSI? Did we discuss that?
so like uh, um,
Your
when we
voice is
were
not clear.
capturing
Yeah.
the risk.
Hmm. Hmm.
So we were referring RSI
Hmm.
there to characterize it as important. We
Hmm.
were uh, uh, looking at severity
Hmm.
and frequency also.
Hmm.
So that is this.
Your voice has lost. We are not able to hear you, Shahrukh.
Hear me now?
No.
Hello.
Yeah. Yes, tell me.
So when you hello, can you hear me?
Are you at some place where there is no network or some fluctuation in network?
Yes, yes.
So first we try to be in a place where there is network because it is creating a lot of disturbance. We are missing your questions in between.
Okay, I will ask later.
Okay, no problem. Any other question? Okay. So now we will come to missing information. What do you think? What could be there in missing information?
Missing information means for data not generated for those patients, we consider that as a missing information.
That is
Like,
correct. Data not generated, or you can say there was some safety profile in pre-animal
yeah,
studies.
not yes, yes.
That is missing information. Okay. For missing information characterization, we cover only one topic, that is evidence source. Okay. Nothing, we don't write anything about preventability, risk group, risk factors, and all those things. We just provide evidence source. Why we provide evidence source? As to on what basis we have considered it as missing information, because we don't have any information. We are still monitoring it. So that's why we provide the source or the evidence source from where, why you have considered it as missing information. As we discussed just now, like if there is some clinical, some non-clinical studies, means animal study was done, and suppose any safety, fertility related, fertility related safety issue was observed in that. So what happened? Animal study was conducted, and in those it was seen that there was some you can say some fertility related was there, or some you can say serious disorder. The special population we the special population which we looked into in our previous sessions were like, what were the special populations, people?
Pregnant lady, lactating mother, and old age people's pediatric.
Hepatic impaired patients,
Renal.
geriatric patients, renal impaired patients, cardiovascular patients. These are all special populations, right? So these population was not included in your clinical trials, and in safety study when you were doing, so you found that there was something related to QT prolongation was being reported in the animal studies. So these will become now or the effect on CVS patients, or this will become your missing information. But for characterization, we don't characterize it on the parameters that we have done in important, identified, and potential risk. We will provide the evidence as to why we have considered it as missing information. In that case, you will go to your RSI. Your source will be mostly your RSI. Source will be RSI. So in RSI, you will go to special population section first, see what is the data that is, means in what population it is provided that the data was not covered. And then you will go to the preclinical section or animal studies section. You will see what safety information is provided. If it is related to pregnancy, then there would be some studies having impact on fertility, and some would be saying that it is passed in via breast milk. In that case, you will take that information from the RSI and put into your missing information for its use in pregnancy and breastfeeding. If it is suppose some CVS related safety information provided in RSI in the preclinical section, and your missing information is used in CVS patients, then there you will provide this evidence that based on this evidence, you have considered it as missing information. And after that, if cumulatively, means in clinical trial, definitely if there is something reported in this, then you can provide it. Otherwise, in post-marketing cases, if you have received some information cumulatively on that missing information, you will provide the same in the same manner that these many cases were reported in this. First, we will take an example itself. Otherwise, you people will confuse every time. Like suppose if I am considering use in cardiovascular patients. This is my missing information. Okay. So what I will do, first I will go to RSI and see on what basis I have taken this as missing information. So basis will be some preclinical toxicity studies in which serious related disorders would be presented, be it QT prolongation, be it hypertrophy, be it hypertension, anything related to that. So that's why we have put it as using cardiovascular. So what statement is provided in that preclinical study, we will paste it here only directly. And then we will provide if any serious related events were reported in CSRs from clinical trials. If yes, then we will provide that information as we are providing for frequency in the above section. And if whatever post-marketing cases were reported cumulatively in use in cardiovascular patients based on the search criteria. And from that, we will provide how many have, what was the outcome, mainly fatal, life-threatening, or anything that we will provide. So this is the only information that we provide for characterization of missing information, nothing else. Okay. Any question?
So for long-term studies, we do not have any source, like we are just putting it in the missing information, right?
Long-term studies, you will have to provide the evidence of why you have considered it as long-term studies. So in that RSI, you will be having some long-term follow-up or long-term effect like section. So you will, there will be some information that since the clinical study was only for the limited period, so it needs to be observed into the what was the long-term effect of. So you can take that information as evidence source and provide it here. And then for long-term follow-up, you would have decided some search criteria, like if the medicine is being used for more than 6 months. So it's the same, you have to provide it here that the long-term follow-up, long-term criteria was selected as this was the search criteria for this. Now done based on that, there won't be any clinical trial information because it is not done. But in post-marketing, if the drug is more than 6 months, then you will fetch the cases. And if you find any cases of the drug being used for more than 6 months, and if there is any safety profile, you will present it here. Otherwise, you will say that these many cases of long-term follow-up studies were obtained from the cumulative analysis, but cumulative line listing, but no cases had any safety profile element. So in this way, you will present the long-term follow-up missing information. Correct?
Okay. So, Amit, I was asking about character. Can you hear me now?
So we will take that question. Just let me finish the today's session first. Anything about this missing information, people characterization? No? Let's complete the section 16 today itself. Section 16.5, it is just for your information. It is called as effectiveness of risk minimization activities. So for your information, just only this section, whosoever asks you, you can tell that this section is a ready to drop in section for us, because we as a PBER writer don't write this section. It is provided to us by the risk management team or the medical affairs team, you can see. And the same is copy pasted here, because we are not writing this section, or we are not analyzing the data available from the risk minimization activities to present into the report. So it is a ready to drop in everywhere. It is ready to drop in. We as a PBER writer are not doing this, and don't indulge into this also that you are writing or something like that. Now let's come to what it is. So what happens, suppose if there is a risk and there is additional risk minimization activity ongoing with it. Like for example, we discuss about teratogenicity. Okay. So teratogenicity is a risk for the thalidomide related drugs, like lenalidomide is there. Then, oh my God, 1 minute. So lenalidomide is there, pomalidomide is there. So these are all the drugs related to the thalidomide class, and they have a common risk of teratogenicity. So for teratogenicity, we have additional risk minimization activity that is ongoing. Like, what is additional risk minimization activity? That the people who are taking this class of drugs, mainly the female, they are not intended to be pregnant during this. Or if they are pregnant, the drug should be withdrawn immediately. So what is our additional risk minimization? We are trying to see that no pregnant lady should get it or get it pregnant while taking this drug. If they are pregnant, we should immediately withdraw it. So our additional risk minimization is prevention of pregnancy, right? So for that additional risk minimization, we have planned something like how we can achieve that. We can ensure that the pregnant ladies are not taking. So first of all, our physicians should be trained on that. They should not prescribe it. Before prescribing, they should have a pregnancy test and all. Second, our pharmacist, when they are dispensing the medication, they should also counsel the patient that they should not become pregnant while taking this medicine, or if they are planning to be pregnant, then don't take this medicine. So these are all additional risk minimization activities done to prevent that pregnancy. Now, suppose if you are doing some activity, then how will you measure its effectiveness, right? Whether the activities which you have planned, are they actually effective or not? So though there would be some criteria to measure that, right? Am I correct, people? Am I correct?
Yes.
Yes, yes.
So those parameters are already decided. So now if I will go deeper, you will be confused. There are process indicators, there are outcome indicators. Process indicators are like, suppose if I have told my doctor, I have trained my doctor. So that training process, that training is my process. So what indicators could be there? That after training, am I getting the pregnancy cases in my database? If I'm getting the pregnancy cases in my database, then my risk minimization activity is not good. I have to change something on my risk minimization activity. In the same manner, suppose if I am if I have trained my pharmacist how to dispense the medication and still I am getting the pregnancy cases. So these are the process and outcome indicators. These are the deep concept for RMP. So no need to worry about it. You can say it is for your understanding that there are additional risk minimization activities. To measure their effectiveness, we have some parameters, but this section is ready to drop in. We take it from RMP people, or you can say medical affairs team who are working on RMP and all those things. So they provide us the effectiveness and we copy paste the information, whatever is provided. We are not involved in writing this section. Okay. Okay.
Okay.
Yes, sir.
So this marks the completion of our section 16 today. It was a long session for section 16 because it is a very important section for PBER. So now we have completed the section 16. So 15 and 16 both are completed now. So now we will move not to section 17 because that we will learn later on. This is also part of medical affairs, but in some clients we write section 17, 18. So I'll teach you section 17, 18, but that could be a later part. We will now move to first, I think, medication error. We will learn medication error in next session. Medication error and off-label use probably. Chalo, any questions till now?
Yes, Amit. So my question was,
yeah, tell me.
in the start of this risk session, we have identified the risk from RSI, right? Like identified and potential based on the causality from RSI only. And then we have categorized it to import, whether it is important or not, right? So we looked at the seriousness and severity and public health impact are like that. So this is the same parameters we are using here for characterization, right?
Right, because that is the only, because we are doing characterization to give evidence that whatever we have categorized is correct.
So,
go ahead.
Yeah. So we didn't look at all the parameters at that time when we are saying like this is important identified risk, this is important potential risk from RSI, right? So that information which we are capturing, so that session was just for introduction, right? This is the whole like proper session on how to categorize the risk based on the characterization.
Yes, at that time I also told you that there we will learn the complete characterization when we are discussing section 16.4. Okay. Now, after you have discussed this section 16.4, do you think that all the information is available in RSI? No, right?
No.
So at the time when you are doing the categorization, I mentioned certain important parameters like seriousness, public health impact, section in which it is available, 4.4, 4.8, causality basically. So these are the basic parameters that you need to categorize the risk. And when you have categorized it as important, identified, potential, and then you have to look for the additional source and everything to characterize it in section 16.4. So it is not that it was just an introduction part. It was just not an introduction part. It was the process that is followed at the time of identification or categorization of risk. Because everything you can't find in RSI, right? In section 16.4, if you will go, then each section means each topic you will not be getting the information in RSI. The same situation is there when you are categorizing the risk initially at the time of KOM. So what are the important parameters? Important parameters are what is the causality provided for that molecule in for that risk in the RSI. Based on that, you have categorized it as whether it is important identified or important potential. Now, whether it is identified or potential. Now, when it becomes important, what is its seriousness? What is its impact on public health provided in RSI? So based on these parameters, you have categorized it, right?
Right.
But characterization is a complete picture of that risk. So that's why it is presented in 16.4, because we look for additional source. But for additional source, we don't look it when we are categorizing it initially. Okay?
So first categorization, then characterization.
Correct.
Also, Amit, I saw one video on YouTube where the person was saying that don't call it as February, because interviewer might know that you are not working in PBR, because always say it as a PBR. So is it right?
So this is just means you can say a fluke, because I might say Pieber, I might say Pibrer, I might say Pebrer. It's fine. But ideally, everybody should pronounce it as Pieber only. Pieber. Not Pebrer, not Pibrer, but Pieber.
Okay.
And that doesn't mean it is a silly thing. Means he might be having some perspective, but it's a silly thing. Like don't say Pebrer or Pieber. It is like Pieber. Everybody says Pieber. Pieber means P. Then how we pronounce it is Pieber. B-E-R. Pieber. But when we write it as Piebrer. P-B-R-E-R. Any other question?
Amit, can you just name that what are the sections basically in which this medical affairs are closely involved in writing this PPP report? Yeah.
Section 16.4 onwards.
From section 16.4 onwards. Okay. Then
Ah,
section 16.4 is it directly closely involvement of that. It's closely involved with this.
medical affairs, but
Mhm.
it
Okay.
is expected from you, because most of the companies we are writing section 16.4, and 16.5 is totally a responsible of that medical. Section 16.5 is totally a responsibility of medical affairs team. But 16.4, 17, 18, 16.4 we tend to write in 95% organization. And then 17 and 17 also we tend to write in 95% organization. But 17, like if someone asks you how medical affairs team, so most of the innovator companies prefer to write section 16.4 onwards by the medical affairs team or the RMP team. You can say mainly the medical affairs team, you can say. But you should say that you are writing, because nobody will take you then if you are saying that no, I'm not writing. Section 16.4 I'm talking about.
Okay, Amit. Amit, apart from that, any other sections? Like you
No,
told about from 16.4 onwards. So apart from that, any other sections?
no, you can't say apart from that any other section that others are writing. If you are not prepared at that time, then you can ask for sales, that sales was being done by other people,
Okay.
sales colleagues or epidemiology section. But it is not recommended, because you should know that. I have shared the introduction template in the chat box. So you guys just download that. Have you downloaded this introduction section? Sorry, introduction template that just we were discussing, no, where I was writing, I have shared that. Are you people to download it? Please confirm.
So downloaded, yeah.
Downloaded, right? Chalo, thank you. And then anything else, or else we can go to our last session. So probably people would be waiting for the person to share his experience from the one who got recently selected, right? Or not?
Yes, yes, please.
Mhm. So I'll close the recording, and let me first close the recording.